Hi, my project is about identifying cell-specific cis-regulatory interactions from single-cell multi-omics data. Here is the overview. In this project, we use the single-cell multi-omics data, which can jointly profile gene expression and chromatin accessibility from the same single cell in a PBMC experiment. And we are going to use this information with some methods to help us identify possible enhancers for each gene, which could regulate the gene expression. So to achieve this, we proposed a method called G. It works as the figure shows. For a specific gene peak pair, the green points are the cells in which the peak in the pair is accessible, while the red points are the cells where the peak is not accessible. Now, if we are considering this specific cell, the first step is to define the gene expression window, which contains cells with similar gene expression as the specific cell. Then, based on the cells in the gene expression window, we will do a binomial test to see whether there are significantly more cells with this peak accessible than the average level. And finally, a cell-specific p-value will be returned for this gene peak pair. We also considered methods based on the Lasso regression model. For the cluster regression, regressors are clustered peak accessibility. As for the adaptive regression, a penalty vector related to gene peak correlation is added. And also for comparison, we considered some existing methods. The first one is the marginal correlation, where we compute the Spearman correlation between gene expression and chromatin accessibility for each gene peak pair, then test against a series of generated background Spearman correlations. Another one is the triple method, which is the published statistical framework for detecting three-way regulatory relationships between a cis-regulatory region, a transcription factor, and a target gene. And here is the summary of the methods. Among all of these methods, G we proposed here is the only one which can identify cell-specific regulatory interactions. The next section is about the results. We used the Phantom project as the benchmark dataset, which has transcriptional regulatory pairs in PBMC cells. Those pairs are used as gold standard in evaluation. AORC and AOPR we used for evaluation are shown in this bar plot for each method here. We observed that G performs better in both terms for the PBMC experiment. From the result of G, we also observe some heterogeneity in regulatory interactions. The result shows that there could be different enhancers regulating the same gene in different cell types, as this figure shows. Lines connecting genes and peaks in different colors indicating that they are detected in different cell types. Also, since we have the cell-specific regulatory interactions, we used these interactions as features of cells and did this hierarchical clustering on cell types and find that they somehow recapitulate the cell lineage. We can see that the cells from the similar cell types are actually grouped closer to each other. We even observe different enhancers for the same gene within the same cell type. Using interactions as features again, we can visualize the cells in this UMAP. For example, in this figure, there are 3 subclusters in the CD14 monocytes, indicating that they may have different regulatory activities. As for discussion, our G method has better performance in the PBMC experiment, and it is able to show cell heterogeneity of regulatory interactions. There are still cons for this method, like it requires large population and longer running time. In the future, we plan to investigate those subclusters in this UMAP for the same cell type and build better model than only use the binomial test. That's all. Thank you for listening.